Thanks so much for having me. I'm going to speak today about virtual patient labs. So this is work that recently came out of my lab, and some of it is still unpublished. So modern oncology is inherently complex, and this is not only because we are looking at patient profiles sometimes over the span of a decade or longer, but also because we are collecting so many different modalities of a single patient. And this is, of course, health records that we are collecting with doctor reports and lab tests. But we are also collecting genomic information, and for especially beyond standard of care patients, where we don't know what to do, we are trying everything. We are looking at drug screens, understanding what would be the most effective treatment for this particular patient. And in the case of oncology, of course, we are looking at tissue data. So we have routine histopathology, but by now we also have those assays that give us this incredible molecular depth into a patient's tissue, spatial omics. And this is simply too much for a single human mind to comprehend. It really requires AI to integrate everything that we have into a single engine, to understand what we learned from one modality and connect it to the other, and in particular to solve this challenge that sometimes even a single modality is simply too hard to analyze by a human without computation. And on top of that, every modality we measure is on a completely different scale, from molecular, cellular, tissue to organ level. Everything needs to be integrated. A virtual patient is an in silico model of an individual's health and disease, and should allow us to really integrate those multimodal, multi-scale longitudinal data into a single representation from which we can ask many different questions, whatever comes to mind. We should be able to forecast and simulate possible outcomes of a therapy that I might be thinking of giving to this patient from all those clinically approved drugs. And then last but not least, we have such a compendium of patients that we've seen in the past. There's so many ongoing clinical trials, but the matching is getting harder and harder because we need to kind of integrate everything we know, everything we measure of this patient in order to make those decisions. This is the task. And of course, a virtual patient doesn't exist, but that's exactly what my lab is focusing on, innovating AI methods that make it possible in order to build such a virtual patient. We focus on 3 different angles of this. First, we need to have a model or multimodal foundation models that allow us to integrate everything we know and can collect of a patient into this single shared representation. From there, we need to use generative AI methods to generate basically the possible outcome a hypothesis might have, a possible outcome of a therapy that we select, possible understanding of how this disease might progress. We can also use generative AI to connect between modalities, because a healthcare system cannot afford to collect everything from every patient, but maybe models that have seen it all might connect how morphology of a tissue actually connects to a molecular readout. And last but not least, it's all not worth it if we can't extract novel mechanistic understanding, better understanding of disease, and then ultimately also guidelines, specific biomarkers that help us to make clinical decisions, and then ultimately also how those systems help decision support and decision-making in the clinic. Throughout this talk, I'm going to talk about those angles very briefly, because that's a very short keynote. And of course, we don't build a virtual patient overnight, so my lab has focused on first building the virtual tissues platform, and so the very first prototype, consisting of the 3 publications listed below, is just released and just in the beta version. So we are the very first ones to build a multimodal foundation model for tissue biology that is trained truly multimodal across histopathology and spatial omics. Of course, we are building on a history of foundation models that have been developed for tissue data in oncology, and those data foundation models have been mostly trained so far on histopathology. Histopathology is kind of an easy modality to work with, because at least it's a data format that we in AI have seen. It's a classic RGB image, and the architectures we use are developed by Meta on Instagram data. They are trained on huge compendia of data, of course, many different cancer types, many different sources, many different indications. And they learn internal representations that we can then use to solve many different tasks at hand. For example, cancer staging, subtyping, or a particular biomarker that might indicate certain prognostic or diagnostic markers. But histopathology falls short in giving us a molecular understanding of the tumor, because it mostly captures morphology. In order to get the molecular depth, we need to look at data such as spatial proteomics that indicate on a subcellular resolution where proteins are located. Problem: this is not RGB data. Here we have actually sometimes hundreds of proteins measured. Every channel is not just a single color, RGB. No, it's the presence of a protein inside the tissue. I just told you foundation models are incredibly data hungry, so we need to train those foundation models on many, many clinical cohorts, but every clinical cohort has different proteins that it measured and recorded. So we need to build foundation models on these incredibly challenging data sets. And for this, we need to start innovating those AI architectures from scratch. The moment we want to build multimodal foundation models, we need to think of this, and then also of the problem that we are dealing with two completely different multimodalities that somewhat still capture the same tissue of a patient. That's the very first achievement of the virtual tissues platforms. It integrates histopathology and spatial proteomics into a virtual tissue state that can now annotate, phenotype, and also virtually stain. So, thinking of every design principles that we back then laid out in this vision paper on the virtual cell, we are using this to really develop completely new architectures that are multimodal and that are multi-scale. They are multimodal across spatial proteomics and histopathology. They're multi-scale because we infuse a protein foundation model to give us every knowledge we have of the protein for which we have the indication of this subcellular localization. Architecture design is innovated from new tokenization systems all the way to the loss function. And it also requires us to collect new data sets because they are not just lying around on Hugging Face. Here we've collected a data set with more than 4,600 patients for which we have multimodal profiles. You can see that compared to the data corpus for which we have histopathology, that's still tiny, but enough to build those models in the first iteration. This is an open source data set, and I'm a small lab. I can only do this because I have received a large grant from the Swiss supercomputer, so we actually trained this on almost 100 GPUs, and this is only the facilitator of this type of research. What can it do? It's one model, many capabilities. So here you can see on the left the data, and our model can segment and cell type pretty accurately compared to, for example, here, a foundation model from Harvard Medical School. But those models are trained seeing all those data modalities, meaning they actually pick up how morphology relates to certain molecular co-localization of those molecules, of those proteins. And you can see our prediction, how those molecules are distributed across the tissue, are pretty close to what we actually measure. And they're better than models that have solely been designed for just solving this one task. But let me rather tell you briefly about what those foundation models enable, what like a pathologist or a clinician cannot simply annotate manually with heavy labor, but now we want to see what capabilities do we add to the spectrum. So we want to be able to forecast how a treatment might affect a patient. We usually do this on cells in suspension, but we want to really capture the complexity of the tumor immune microenvironment in order to make this possible. Our virtual tissue platform is the best way because it gives us already a representation of my virtual tissue. Now I can develop perturbation simulators that forecast how those tissues might change upon perturbation with a potential drug, and I can screen all of them and then ultimately read out the biomarkers that are relevant for clinicians to make then a decision of what is the most effective treatment. We call this a biological world model because here we really have a state model that encodes this complexity of human tissue, and an action model that kind of understands how to navigate between those different tissue states. For this, we got the 8i 2050 grant to further develop this over the next couple of years. What is interesting for us is to really extract signatures that let us decide if a patient is a potential candidate for a clinical trial or for an immunotherapy, to really have some understanding, is this going to be a successful therapy, yes or no? Current biomarkers are mostly manually handcrafted or depend on fairly simple ratios. But here, we show you that the virtual tissue platforms actually allows us to extract what we call foundation model-based biomarkers. We have here a clinical cohort for which we actually also have samples after treatment response. So we can use this to understand if our biomarkers are predictive for the after state. Again, you see here a virtual tissue representation. And in fact, you can see already from the learned state, it learns cellular identities in particular spatial arrangements and ultimately signatures that are going to be highly predictive for response to immunotherapy, yes or no. And this, on the gray, very right, are the current clinical stratification schemes. The dark blue is the original Nature study that proposed in an entire publication a handcrafted biomarker. Those handcrafted biomarkers don't translate to the new clinical cohort if I have a new patient in a different context. Those foundation model-based biomarkers do. Here, on a completely independent clinical cohort, we can take the same biomarkers and get a stratification into low, high, and medium risk towards, again, immunotherapy. So this is super interesting and exciting because it really opens a completely hypothesis-free angle of what are biological markers that derive decision-making in the clinic. Last but not least, those foundation models should be active companions in a clinical setting, meaning that they also should allow us to optimize the processes for the entire healthcare system. Do I need to really develop and measure all the data I possibly collect for a patient, or is it enough that for some patients I'm only collecting a minimum amount of diagnostic and prognostic tests because the model is able to complement the rest? How to make such decisions? Again, we take the virtual tissues platform, but now we have a representation that is good enough for many different downstream tasks. We developed a new test time training system, a test time inference system that allows us to kind of represent what the model knows about its internal state. And based on this, using an RL framework, kind of sequentially collects more and more modalities until it knows itself it has enough information to do very well on all different downstream tasks. We call this ECHO-K, and this is really a task-agnostic way of accumulating modalities to then really do well on a minimum assay I collected for a patient for all the downstream tasks I care about. You can see here, for example, for cell typing, the model sequentially asks for more and more protein markers, but it stops at 8 because it's simply enough to do all the downstream tasks we care about, and it doesn't need more. What is the overall vision, right? So we are building those models to ultimately assist clinicians in the clinical setting, to really somehow support analysis of patient samples. And so we recently had a paper at NeurIPS to understand how can those foundation models that we built be integrated in with molecular tumor board agents. And now we are closely working more and more with clinicians to understand how could something like this possibly be deployed in a clinical setting. And with this, I would like to thank my collaborators, in particular my lab, of course, at EPFL, but the clinicians at University Hospital in Zurich, Andreas Wicki, and the University Hospital in Geneva, Olivier Michelin, with which all of this work was in fact co-developed. Thanks so much.